Howdy folks, welcome to chapter 15, sure our pen works. This is hearing, second video in hearing, so clearly we're right here with the ear. Just a review of neurotransmitters and synapses. So here's our neuron of interest, is the black neuron. These black spikes obviously are all dendrites. These ports of entry into the neuron are channels. So we have here's a blue channel, we have a red channel here, another blue channel in the axon hillock, and then a black channel here. And then these green things are neurons, and then the little lines sticking off them are dendrites. And then here's the axon. Here's the axon terminal of the green neuron. So the green neuron sends an action potential down its axon to the green neuron's axon terminal, releases a neurotransmitter. Neurotransmitter binds to this channel. So this is a ligand-gated channel. Remember, we talked about this earlier. Ligand-gated channels, when they bind their neurotransmitter and they open, whether positive or negative charges come in, it will always be a graded potential. So if positive charges come in and you measure the membrane potential in this area, because positive charges are coming in, the resting membrane potential over here will change to a depolarized graded potential. So positive charges come in, you get a slight depolarization and then a repolarization back to resting membrane potential. But the point is, here in the neuron's cell body and their dendrites, that's where you do graded potentials. If enough of these positive charges build up, enough of these ligand-gated channels are open, positive charges come in, and they come over here to the axon hillock, they can activate these voltage-gated, so not ligand-gated, but voltage-gated sodium channels, and then sodium can rush in here as usual, and you form an action potential. And then here's a graph of an action potential. So the resting membrane potential is here. Positive charges are drifting from the cell body over to the axon hillock. If enough positive charges make it to the axon hillock here on the neuron, and the membrane potential depolarizes to minus 55 millivolts, that's the threshold potential, then you can fire an action potential. And then that action potential will travel down the length of the axon till it reaches the axon terminal here. If the black channel, say over here, let's say a neurotransmitter binds to the black channel, and this time instead of positive charges, you have a bunch of negative charges coming in. What that's going to do to the membrane is hyperpolarize it, but it's still a graded potential, but this will be a hyperpolarizing graded potential. So in order to understand the special senses and how they transmit signals, we have to understand that neurotransmitters in the cell body and dendrites of a neuron will cause a graded potential. Action potentials only happen in the axons. Okay, so I think we went over this in the last slide, but here's your ear. So here's your outer ear, here's the canal, here's the tympanic membrane, and then this is the inner ear, so they enlarged that cartoon. This stuff out here, this is all temporal bone. And then in the top part of the cartoon, it shows the semicircular ducts in semicircular canals. So here, this tan stuff, these are the semicircular canals. So they're made of bone, and then the ducts are within the canals. And you have anterior semicircular duct in a canal, posterior semicircular duct in a canal, and then this is the lateral semicircular ducts within the semicircular canals. And then we talked about this last time. This is the crista ampullaris in the ampulla of the semicircular canals. Here's another one. Will detect equilibrium. And then this whole area, this is all the vestibule. And then there's these two purple items, and they are called maculae, and within these maculae live the utricle and the saculae, and they're involved in equilibrium. So we'll talk about those when we do equilibrium. And then here's the nerve. So here's the facial nerve cut. Here's the vestibular nerve. So it's going to go here to the vestibule. And then here's the cochlear nerve that will go to the cochlea where we do our hearing. This is the stapes covering the oval window, and then this piece right here is the round window. And then the cochlea, it spirals around. So there's a cochlear, there's the cochlea, and then this blue stuff is the cochlear duct within the cochlea. And then this purple stuff is the spiral organ of Corti, and that's where we'll do all our electrophysiology to transduce a sound wave into an electrical signal that can go to the brain and tell you you just heard something. So this is kind of a cool cut. So it looks like this is kind of a vertical section and then kind of a trans section here, and then they pull this piece out, and then you look inside and you can see the cochlear nerve coming in, and it's basically innervating all these little structures. So these are all the organ of Corti. We'll look at that in a minute in more detail, but these are all organs of Corti being innervated by branches of the cochlear nerve. And the organ of Corti sits on this thing called a basilar membrane. And the organ of Corti is in this duct called the cochlea duct, aka the scala media. And then next to the scala media you have the scala tympani, so this duct, and the scala vestibuli, that's another duct. So the cavity of the cochlea is divided into these three chambers. The scala media, so we just looked at that. The scala media is also called the cochlear duct. It contains, the fluid in it is called endolymph, and the organ of hearing is called the spiral organ of Corti. The basilar membrane will support the spiral organ of Corti, and it also contains sensory receptors for hearing. So like I said, that's where we do our hearing, is in the spiral organ of Corti. Then within the cavity of the cochlea, the next chamber is the scala vestibuli. So that's this guy right here. And it actually begins at the oval window. And it's superior to the cochlear duct. And it contains not endolymph, but a fluid called perilymph. And then last chamber within the cochlea is the scala tympani. It's inferior to the cochlear duct, and it also contains perilymph. And the scala tympani ends at the round window. So, and it's over here. Okay, in this cartoon, I'm not sure, I think in this cartoon this is the round window and this is the oval window. So remember, the stapes sits over the oval window, and the stapes is going to move back and forth with the movement of the tympanic membrane, and it's going to start fluid, that endolymph and perilymph. It's going to start the fluid moving in your ear, and then that's going to excite sensory receptors so that you can hear. So here's a better cartoon that shows you the organ of Corti. This is the cochlea. So remember, superior, well, let me back up. Here's our anatomy of interest. So this thing I just circled is the cochlear duct, aka the scala media, and there's endolymph in here. So all this blue stuff, this is all a fluid called endolymph. And then superior to the cochlear duct or the scala media is the scala vestibuli, and it contains, the fluid in here is perilymph. And then inferior to the scala media or the cochlear duct is the scala tympani. It also contains perilymph. But here's our hero, the spiral organ of Corti, right here. This is the spiral organ of Corti. And On the top of the spiral organ of Corti is this thing called a tectoral membrane, and it's a stiff membrane, and we'll look at that in more detail in a minute. But then here is the ganglion, where you have synapsing with the fibers of the nerve that will take information from the spiral organ of Corti to the brain. So the floor of the cochlear duct, that's here, is composed of this thing called the basilar membrane. The basilar membrane supports the organ of Corti, but the basilar membrane can actually move up and down. So it's really important to know. The basilar membrane can move up and down depending on the sound waves that are traveling through the endolymph and paralymph. Okay, this is a bigger cartoon of the spiral organ of Corti. Again, here's the tectoral membrane up top, and you can see that tectoral membrane is being pushed on by these little hair-like projections called stereocilia. And these stereocilia are part of these outer hair cells. So this is an outer hair cell. Here's a whole outer hair cell with its stereocilia. Here's the outer hair cell nucleus, and you can see that fibers of the cochlear nerve are making contact with this outer hair cell. And then these guys out here are support cells. They don't have stereocilia on them. There's also these cells called inner hair cells. So they're inside, or they're inner hair cells. They also have a stereocilia, and those stereocilia will bend with sound. So the outer hair cells and their stereocilia will stiffen the basilar membrane. So this is the basilar membrane. The whole unit is sitting on the basilar membrane. The outer hair cells will make it stiffer, and the inner hair cells, their stereocilia will bend with sound waves traveling through the endolymph or paralymph. And this is an afferent nerve fiber. So the afferent, this is a sensory afferent neuron, fiber of a sensory afferent neuron. And then these are the fibers of the cochlear nerve. So remember, a nerve is just a bunch of axons running together in the peripheral nervous system. So this is a scanning electron micrograph of the spiral organ of Corti. Here's the outer hair cells with their stereocilia, and then here's the inner hair cells with their stereocilia. So I think this would be an inner hair cell, and here would be the stereocilia of the inner hair cell. All right, so let's talk about some physics, shall we? Properties of sound. Sound is a pressure disturbance produced by a vibrating object. So probably everybody's played with one of these before. It's a tuning fork. Okay, so here's the rubber mallet. You hit it, and then these little blue balls, those are all molecules in the air that can vibrate. So you hit the tuning fork here, and this part of the tuning fork will move that way and push these air molecules in this direction. And then this guy, the other part of the tuning fork, will move, and molecules will be pushed in that direction. And it shows you the arrows out here. So basically this tuning fork is moving back and forth, and you have areas of where the molecules in the air are compressed. So this is called compression, and then where they're spaced out is rarefaction. So sound waves travel compression, rarefaction, and then all these molecules will get compressed again, and then rarefaction. Anyway, and then through a series of movements, that sound, if you're in range, will hit your tympanic membrane. And this just kind of shows how sound waves can travel out in all directions. So from where the sound initiated at the tuning fork sound travels in all directions, moves outward in all directions. Okay, now, so we talked about you hit a tuning fork and then you get an area of high pressure where the compressed molecules are. That's compression. And then where there's low pressure or fewer molecules, that's rarefaction. And then you can associate that with a graph of the wavelength of these waves. So here's air pressure on the y-axis, here's distance on the x-axis. So the areas of compression are associated here with a peak in the wave, and then you come down and you hit a trough of the wave here. That's associated with rarefaction or areas of low pressure. Then you come back here, get high pressure, molecules are compressed, low pressure, molecules are not compressed. And then, so the tip, the peak of the wave is called the crest, and the base of the wave is called the trough. The distance from the trough to the crest of a wave is called the amplitude. And then, like I said, the distance from one wave, the crest of one wave to the crest of another wave, that's the wavelength. Okay. So properties of sound waves. So let's look at this. Let's look at A first. So we've got pressure on the y-axis, time on the x-axis, and it tells you the key up here. The red, these are high frequency, short wavelength waves. That gives a high pitch. So you guys probably know the voice has a pitch. You can have a high-pitched voice or a low-pitched voice. If you have a high-pitched voice, then you're transmitting these high-frequency, short wavelength waves. So here's high frequency because from wavelength to wavelength is pretty short. So red crest to red crest, as opposed to, look at this, blue crest to blue crest. It's got a lot longer wavelength. So when you make a sound that's low frequency, you'll have a low-pitched voice and you'll have a long wavelength. Frequency is perceived in our brains as pitch. Sound, amplitude is perceived as loudness, a loud sound. So if you make a high amplitude wave, like this blue one, the amplitude will translate into a loud sound. If you have a low amplitude wave, that will translate into a very soft sound. So here's just some definitions. The frequency of a wave is the given number of waves that pass a given point in a given time. Pitch, that's how we perceive different frequencies. Amplitude is the height, so the height of the crest. That gives you the intensity or loudness of sound. And the normal range that we can hear in is 0 to 120 decibels. And I think this is a logarithmic scale. Okay, so now we'll look at transmission of sound to the internal ear. So we're on step 1. So this is our sound wave coming in. So this sound wave here's its crest, here's its trough. From here to here is its wavelength. The height is its amplitude. So these sound waves have all these physics characteristics, but the sound wave comes in and strikes the tympanic membrane, and the tympanic membrane starts to vibrate back and forth, back and forth. Because the malleus of those 3 inner ear muscles is attached to the tympanic membrane, that means the malleus is going to start moving back and forth in time with the sound wave. Malleus is connected to the incus, and it will move back and forth. The stapes, the third bone here, the stapes will move back and forth. So as the bones vibrate or move back and forth, the initial pressure that came into the tympanic membrane, that's amplified. So that initial pressure of that sound wave gets bigger. And then because the stapes is covering the oval window, which is like a membrane, the oval window can move back and forth and back and forth. And the oval window is the port into the scala vestibuli, filled with perilymph, so a fluid. So now, that initial sound that came into your ear, moved the tympanic membrane and the bones, is translated to moving fluid or perilymph within this structure called the scala vestibuli. So now you set up the waves in the fluid called the perilymph, and it travels. Sounds with frequencies below hearing, so that we can't even hear, travel through the helicotrema. So that's, if you unwound the cochlea, the tip there, that's the helicotrema. And no hair cells are excited, so you don't hear anything. If a sound is in our hearing range, it'll move through the scala vestibuli up here, and well, it'll move, well, let's look at 4B. It goes through the cochlear duct, so that amplitude, that frequency of sound wave will indent here on the cochlear duct, and it will start to vibrate that basilar membrane, and it will deflect hairs, those stereocilia, on the inner hair cells, and then those inner hair cells are going to do some biochemistry to tell the cochlear nerve that there's a sound present. Okay, so let's take a look at this cartoon. Here's the tympanic membrane. Here's the frequency of the sound wave coming in, moves the bones. The bones, the third bone, the stapes, will start moving back and forth, and that will make the perilymph in this port, it will start that fluid moving. So, and now let's come over here to the left. So in this cartoon, they've unwound the cochlea. Okay, so it's just straight out. It used to be wound up kind of like a snail, but now it's straight out. So if you have a high frequency sound, so this is, check it out, this is high frequency, because look, there's a whole bunch of waves passing per unit time. That's a high frequency sound. Then those sounds, those waves, will displace the basilar membrane, which is in here, will displace the basilar membrane near the base of the cochlea. So remember, this is the tip of the cochlea, or the helicotrema. Here, this is the base of the cochlea, where it starts. So if you are a high frequency sound, you're going to displace that basilar membrane near the base of the cochlea. If you are a medium frequency sound, so check it out, this is medium frequency, then you're going to displace the basilar membrane inside here in the cochlear duct near the middle of the cochlea. If you are a really low frequency sound, so look at the distance from wave to wave, wave crest to wave crest. This is a real low frequency sound. You're going to displace the basilar membrane near the apex of the cochlea. So different sound frequencies cross the basilar membrane at different locations. And then let's look at this cartoon. It says fibers of the basilar membrane. At the base, the fibers in the basilar membrane, it's short and stiff, and they will react to these high frequency sound waves. If you're in the middle of the basilar membrane and you have a medium frequency sound, that's where you'll displace the fibers of the basilar membrane. And then at the apex, so out here, you have long floppy fibers of the basilar membrane. So the low frequency sound will displace those fibers. So the sounds in the hearing range go through the cochlear duct. So those sounds, it's pressing down here on the cochlear duct, pressing down here on the cochlear duct, and the sounds in terms of their waves is pressing down here on the cochlear duct. And that when they press there, they're going to vibrate the basilar membrane, and that'll be commensurate with the frequency of the sound wave. And then we talked about this. There's near the oval window, so right here, there's shorter basilar membrane fibers here, and they resonate in response to high frequency wave. Near the apex, you have longer basilar membrane fibers, and they resonate with low frequency waves. And then here's the cartoon of the fibers. So here's the basilar membrane, and remember and then here's the scala vestibuli with the perilymph in here, and when that fluid is moving, it's going to cause the basilar membrane to move up and down. And when the basilar membrane moves up and down, those fibers are going to move up and down because they're attached to the inner hair cells and the outer hair cells. Okay, so we've looked at this. Let's see, afferent fibers of the cochlear nerve, that's afferent, so those are sensory afferent fibers. They coil around the bases of the hair cells. So it doesn't really show it coiling, but these sensory afferent neurons are coiling around the base of the cochlear hair cells. The outer hair cells, so these guys out here, these are the outer hair cells, and then this is an inner hair cell. The outer hair cells and their stereocilia, those are these little hair things right here, they protrude into this tectoral membrane that's real stiff. And that, because that, you've got that stiffness between the stereocilia and the tectoral membrane, that's going to make the basilar membrane stiff. But when you get a sound wave moving through the perilymph, then that basilar membrane is going to move up and down according to the wave in the perilymph, and the stereocilia on the inner hair cells will bend. Now, I don't have a cartoon of this, but you'll have to use your imagination. But when you bend these stereocilia, they have ion channels in them. So, and they're mechanically gated ion channels. So when those mechanically gated ion channels open, because the stereocilia are bending, then you'll get a graded potential here in the inner hair cell. And if there's enough bending and enough ions coming into the inner hair cell, then the inner hair cell will release a neurotransmitter here onto the sensory afferent neuron, and then the sensory afferent neuron will send an action potential through the cochlear nerve back to the brain, and you'll say, oh, I just heard you say that. Okay, auditory pathways to the brain. So this looks like it's a frontal section through the brain. So all this, remember, is cerebral cortex. All this white matter, those are just tracts of axons from neurons. And in here it says the medial geniculate nucleus of the thalamus. So this purple thing is the thalamus. Nucleus, remember, is just a collection of cell bodies and also axon terminals in the central nervous system. The primary auditory cortex is in the temporal lobe. So this is all primary auditory cortex in the temporal lobe. That's where you hear sound, so it's the auditory cortex, right? So you hear stuff. And then there's these other pieces. Here's a synapse in the inferior colliculus. Here's a tract, the lateral lemniscus. Here's a superior olivary nucleus. So you've got some synapses here. Cochlear nuclei. And this piece here, this is this cross section through the midbrain. Here's a cross section through the medulla oblongata. Here's the vestibulocochlear nerve, so it's taking information from your equilibrium apparatus and your hearing apparatus. Here's the spiral ganglion of the cochlear nerve, and then this is a bipolar cell. So basically, when you, let's see, what do I have in here? So remember, the inner hair cells are here. When the sound, well, it shows here, when vibrations come through the perilymph and start the basilar membrane moving up and down, these inner hair cells, stereocilia, will open mechanically gated ion channels. Ions come in, you get a graded potential in the inner hair cell, and then if it's great enough, you'll get an action potential that will send through a sensory afferent neuron through the axon of that sensory afferent neuron, where it will synapse here with a second neuron in the spiral ganglion of the cochlear nerve, and then that information to get to the primary auditory cortex in the temporal lobe will travel here to the medulla. You synapse. Got some synapsing here. You've got some synapsing up here in the thalamus. Then finally, that information about what you heard goes to the primary auditory cortex in the temporal lobe. So the cochlear nerve to the medulla. So the vestibulocochlear nerve to the medulla, and then it goes to this inferior colliculus, and then it goes to the thalamus. Then from there it goes to the primary auditory cortex. And it says here auditory pathways decussate so that both cortices receive input from both ears. Okay, impulses from specific hair cells. So here's our inner hair cells. An impulse, so remember that's an action potential, are interpreted as specific pitches, so high pitch, low pitch, medium pitch. Loudness, the way you detect loudness is detected by an increased number of action potentials. So when these inner hair cells experience really large deflections, so they're moving up and down in that basilar membrane, you'll detect through all the electrophysiology and biochemistry, you'll detect a really loud sound. Homeostatic imbalances of hearing, there's conduction deafness. So if sound is blocked, you can have, that can be the result from earwax. So you just have a bunch of earwax built up. Your eardrum could be perforated or torn, so it can't move back and forth, and you can't send that sound wave information to the bones of the inner ear, and then they can't send that sound wave information to the perilymph. There's sensorineural deafness, damage to the neural structures. So any of these neural structures, these blue neural structures, damage at any part of that pathway could result in deafness. Tinnitus or tinnitus is ringing or clicking sound in the ears. Even though there's no auditory stimuli, you could have cochlear nerve degeneration, inflammation of your middle ears. It's also the side effect of aspirin, tinnitus. And then there's the syndrome called Meniere's syndrome. It's a labyrinth disorder that affects the cochlea. So remember, this is all bony labyrinth. So here's the cochlea. It's all inside this bony labyrinth. And there's some problem within the labyrinth that affects the cochlea, and also these semicircular canals with equilibrium. And the result is you get vertigo, you're nauseous, vomiting.